Our next speaker is Dr. Alison Van Eenennaam, and I was practicing all day to be able to say her last name. You know, I talked to her this morning, and we were joking about having accents and having last names that people butcher all the time. So, Dr. Van Eenennaam is from the University of UC Davis and is an extension specialist in animal biotechnology and genomics, and will be talking today about genetic and management approaches to reduce bovine respiratory disease. Welcome.
Thanks very much, and good on you for staying till the bitter end here. The reason we swapped computers is I'm recording this because I like to put my presentations up on my website, and so that was the reason for doing that. So as you can probably see by my Twitter handle there, I work a lot in the beef industry, and I'm going to talk today, however, about a project that I've been working on, the Bovine Respiratory Disease Coordinated Agricultural Project, which is a collaborative project that involves both beef and dairy industry. And so I think the previous speaker gave me a nice introduction to the fact that bovine respiratory disease is a real issue in the dairy industry, and as a nutritionist he looks at it from a nutritional perspective, and as a geneticist I'm interested in it from a genetic perspective. And so I'm going to go over a little bit of the research that this 5-year grant is going towards, and then spend the last part of the talk discussing dairy calf management practices as it relates to nutrition, and kind of interestingly enough as it related to the welfare talk earlier in the day, for those of you that were attended that, to have a look at what practices dairymen in California are using with regards to their young calf management. So what really prompted our interest in this is that despite incredible amounts of research that's gone into trying to reduce the incidence of bovine respiratory disease, we have pretty much the same levels of incidence and mortalities that we've had for the past 30 years. And so despite improved vaccines and broad-spectrum antibiotics, we really haven't made much progress. And so what we were interested in doing is having a look and see, is there any approaches we can take to develop genetic tools to help address this problem. So for those of you unfamiliar, bovine respiratory disease, or BRD, is a significant cause of morbidity, mortality and economic loss, and antibiotic use in both dairy and the beef industry. It's associated with many pathogens. Typically viral comes in first and then bacteria come in later, exacerbated in times of stress, particularly in dairy calves in the pre-weaned period. In beef cattle it tends to be more associated with shipping fever, when those animals are going into the feedlot and getting mixed with new animals. And overall it's the largest natural killer of cattle in the United States, and in the dairy it's taking about 18.1% of all pre-weaned dairy heifers are estimated to experience respiratory disease, and it kills about 22.5% of the mortalities in unweaned dairy heifers and 45% in weaned dairy heifers. And there's economic costs associated not just with the treatment of disease, but I think much longer term effects in terms of the thriftiness and the health of that animal that carries over into its lactation. And I think that that's was echoed by the previous speaker, and the importance of trying to prevent the animal from getting sick in the first place is really what spurred us to develop this 5-year grant, that the overall objective of which is to really utilise the power of some of the new genomics tools to see if we can start to tackle some of these more difficult to manage disease traits. And that's what we're trying to do, and I'm going to discuss a very large field study that we've conducted trying to identify regions in the genome that are associated with animals that are getting less bovine respiratory disease. And it also has a large educational component and an extension component that's associated with this, both in terms of management, because it is both a genetic and a management disease, and then also training graduate students and undergraduates. So that gives you some kind of a feel for the scope of the research team that's behind it. It's led by Dr. Jim Womack at Texas A&M University. There's the research lead is Holly Nybergs at Washington State University. I'm the extension lead located in California, and then Milt Thomas and Robert Hagewort, who we heard earlier today, are kind of leading up the educational components of the grant. So If you attended the genomics sessions yesterday, you probably heard a lot about genomic predictions for traits that we already have good information on, and so I think we've had tremendous progress in terms of doing DNA tests on animals to predict traits like milk production and the like. But really the kind of the value of genomics is that they're very useful for traits that are difficult or expensive to measure. And I think disease is one that falls very clearly in this. So we really don't want to expose the animals to determine which ones are susceptible and which ones aren't. We'd much rather be able to genetically test them. And so they're useful genomics to measure traits that can't be measured until later in life, like carcass characteristics. And they're currently not selected for because there are no routine measurements, and I think that's such an important point here. The only reason genomic predictions work so well in the dairy industry is we had an amazing database of phenotypes recorded by the DHIA, and we had a semen tank full of DNA from those well-proven bulls. We had what's called the training population sitting at the USDA offices and just needed to do the genotyping and then match up which regions of the genome were associated with positive effects on milk production or whatever we had records on. If you don't have records, we can't do that. If you can't measure it, we can't get prediction equations, and that's really the problem with some of these disease traits. And I understand yesterday the discussion was we would like to start having genomic tests for some of these disease traits. Well, we can't do that until we have big populations that we can use for training. So the availability of these SNP chips, the 50K SNP chip you're probably familiar with, has given rise to really this opportunity to select for traits that we really couldn't do previously, but we realise we need to have thousands of observations in order to have a sufficient population size for us to detect which little regions of the chromosome are associated with animals that are less susceptible to disease. And so when we're talking thousands of individuals, we really have to go to field data sets. We have to go to commercial collaborators because no one university is going to have anywhere near the numbers that are needed to get the sufficient numbers. But unfortunately, field data, especially as it relates to disease, is very noisy. And by noisy, I mean you're not very accurate at determining which animal is sick and which animal is healthy. And there's a couple of reasons for that. One of them is, how do you determine whether an animal has BRD? Is it just someone looking at the animal and thinking they look a little bit depressed? And maybe you've got the calf person out there who's maybe getting an incentive not to have sick animals, and so he'll be very rapid to call an animal sick and treat it to avoid potentially having an animal die. And also you'll have apparently healthy individuals who in fact are susceptible but never got exposed to the agents or the viruses and the bacteria, and so they look like they might be genetically superior, but in fact they've never been exposed. And so all of that adds what's called environmental noise to the problem, which basically decreases the heritability of disease resistance. And heritability is, of course, how much genetics explains a trait. And traditionally, disease is a very lowly heritable trait because there's so much environmental influence and because it's very difficult to get this precise definition in there. So, you know, no one said we're going after the low-hanging fruit. So with genomic predictions, we've already got the low-hanging fruit. That's what your PTAs are that you have currently in your genetic evaluations. Recently, big grants have been funded to try to start getting the higher hanging fruit, feed efficiency, reproductive traits, and then finally, of course, disease-resistant traits. And so there's a very high risk but a very potentially high reward, because the presence of genetic variation to disease resistance, I think, in conjunction with increasing public concern about the use of antibiotics, and of course there's a lot going on at the federal level as it relates to that, makes selection against disease a very appealing project for geneticists to go after. And so that's really was the reason that we pursued this particular project. And we have made success in targeting against disease. So you're familiar with somatic cell scores and the fact that that's been utilised to select against mastitis, and once you implement that and put that into your selection criteria, so it's part of the net merit index, appropriately weighted versus other traits, we can start to make improvements in disease, even though it's often inversely correlated with traits that are economically important, such as milk production. Chicken breeders have been doing that for years with various diseases that are of importance to their industry, and recently genomic efforts have resulted in a very large effect loci that's associated with something that's going through the pork industry at the moment, the PRRS. And so there is some precedent that we can make improvements against disease by including it in selection. So the real meat and potatoes of the BRD CAP project is developing these large case-controlled field data sets that we can use to determine which regions of the genome are associated with animals that are not getting sick. And so we've basically developed these large data sets. There's 2,000 in California, and I'll discuss in detail what they were, where we had 1,000 cases and 1,000 controls, and then there was similarly 1,000 animals in New Mexico that Robert Hagelwart was involved in collecting, where we had 500 cases and 500 controls. And importantly, we're following all of those animals through their lactations to have a look. There's economists on the team having a look at what the impact of getting disease versus the controls is on those data sets. All of them are genotyped with the 800K SNP chip, so that's the 50K, but 800, because we wanted to have very closely spaced markers throughout the 3 billion base pairs that make up the genome. And we've also done very thorough characterization of which pathogens were associated with the animals so that we can look for genetic by pathogen interaction. For example, are there regions of the genome that help you be more resistant to Mannheimia hemolytica, for example, or something like that. And so one of the really important aspects of this was trying to get a very objective measurement of whether an animal was sick or not, because we wanted to not have a subjective interpretation, but rather use very precise definition of what an animal was. And so where we did this study was on a large 70,000 head capacity dairy raising facility in California. It was done, led by Dr. Terry Leyenbauer at Tulare and Sharif Ali, and Jessica Davis was an intern that was involved in collecting data. And basically what we did was every morning walked the pens looking for animals that were sick, and the way that was done was we used Sheila McGurk's calf respiratory scoring chart, which I'll discuss on the next slide, and basically it looks at temperatures, eyes, ears, nose and cough, and gives it a score, with more abnormal animals getting a higher score. And in our case, they had to have a score of greater than 5 to get enrolled. We also at that time collected blood, and once an animal was enrolled, we collected blood and did a deep pharyngeal nasal swab. But importantly, we took a calf that was adjacent or very close by to the sick animal to be our control animal. That is, an animal that's likely to have had similar environmental exposure to the sick animal, but is apparently healthy according to the scoring chart. And so I'll show you what the scoring chart looks like. So basically, the way this chart works is if you get a 0 score, you basically have normal rectal temperature, normal cough, or no cough at least, and normal looking eyes, ears, and nasal discharge. The more severe the signs are, you start to go up to 1, 2, until over in 3 there, you have, very high temperature, a spontaneous cough, bloody nose, mucky eyes, and a head tilt. And those 3 cards on the far right of that screen, I think pretty easily, I probably don't even need a scoring chart to say that those are animals that are sick with bovine respiratory disease. And so they had to have a 5 or greater on that particular chart for them to qualify as a case. And so this is just examples of collecting blood and doing the deep pharyngeal swab collection. And the deep pharyngeal swab is really important because we didn't want to sample what bacteria were in their nasal passage. We were more interested in what was infecting their lungs. And so if you look at how a deep pharyngeal swab is done, it's basically a guarded swab that passes back into the deep pharynx, the pharyngeal cavity there, and has been found to be a less invasive approach than doing a lavage of the lung, but it still gives you quite a good representation of what bacteria are infecting the lung. So that was done for the cases, and then the controls were scored the same way. And it was really important that we were able to do this because we had an objective measurement of cases and controls of animals that had a similar environmental exposure, with the hope being that we'd really be able to hone in on the genetic attributes of being susceptible versus sick, rather than different environmental impacts. And so what we're trying to do effectively is try to raise the heritability of disease resistance by decreasing our environmental variability. And that's basically what we were successful in doing. This is just kind of to give you a visual representation of what this looks like. This is the 29, I guess 30 fell off the page there, but the 30 top sires in terms of AI sires that were represented in our data set. And so this data set was just calves that came in from a great number of dairies, and of course, because we have 50k genotypes on all the bulls, we could tell who sired who. And you can see here, of the bulls that had 10 or more progeny in our data set, I was interested in just having a look, well, are there cases where, you know, if genetics has nothing to do with it, we should have about 50-50 case controls in each particular bull. And what you see here in the red and the green line is certain bulls where you've got a much higher than predicted proportion of animals that are sick, in the case of bull number 3 there, and in the case of bull number 12 there, you've got a much higher than predicted and statistically significant cases of healthy animals. And so looking at that data, you know, you might think number 12 might be a bull that you'd be quite interested in using, and maybe less likely to use number 3. Of course, these are going to be fairly highly used AI bulls for them to have this sort of representation in the data set. And so this was kind of a sign that we were actually getting some heritable differences. And what the preliminary results show is basically, in order to get the data set we put together of 1,000 cases and 1,000 controls, we estimate we probably screened about 200,000 calves, just walking up and down looking for that, the one that fit our case definition, and then getting a control that went along with it. And the calves that were identified as cases were significantly associated with positive cultures in the viruses and bacteria that are associated with BRD, which was satisfying and suggested we were doing the right thing there. Just of interest is they were all DV negative. But what the real take-home message is that when the genetics researchers analysed the data, and this is work that's done by Holly Nybergs, Chris Seabury at Texas A&M, and Jerry Taylor at Missouri, they did it 3 different ways. We basically came up with heritability estimates of about 21%, which is really very good for a disease trait. And we were explaining about 20% of the variation in BRD incidence, which is pretty good. So we've now identified regions of the genome that are associated with less susceptibility to BRD, and currently what's happening is work at Texas is trying to hone in on ideally the causative gene that's associated with that variation, but try to get closer to where that variation is, so that we can get tests that are able to track that very efficiently. And we're also working with Kurt Van Tassel, who's at Beltsville Lab, the USDA, to start implementing that into PTAs, or genetic predictions. And of course, in order to do that, we'd ultimately need to include the trait of BRD in net merit index. And so, of course, then that would have to be appropriately weighted according to the value of that trait relative to all the other economically important traits. And so for a real implementation into the industry, that's kind of what we think would have to happen. And I think it's interesting to watch the evolution of the net merit index over time. And you can see back in 1971 there were only 2 traits in the net merit index. There was milk yield and fat. And as time has progressed, we've started to incorporate more and more traits of economic importance to the dairy industry into the net merit index, until in 2014 you can see, in fact, milk gets a -1 rating as it relates to yield. Of course, it's positively correlated to both fat and protein, but there's a large number of traits, and I can see as we develop genomic tests for more traits that this will become even more complex and more representative of what really is an optimal animal for the industry, rather than just selecting on 1 or 2 traits. And so I think there'll be an evolution in the index as we get into this. So I'm going to move off the BRD cap now, and I guess just out of interest and curiosity, when I first started this project, I typed in BRD cap, and there was a BRD cap that you could buy. And so I was kind of curious what BRD stood for, and I looked up this website where you could buy this hat and t-shirt, and it's black rifle disease, which is something like an obsessive compulsion to utilize semi-automatic black rifles. So I just want to be clear, the BRD cap I'm speaking about is not associated with that particular BRD cap. So, but I think it is an interesting time to start having a look at how we might be able to use records on health traits to develop genomic selection tools. And there's an interesting paper came out where dairy herd records that producers had kept in databases in computer programs have been used and trained to develop genetic predictions for a large number of different health traits that are shown on this slide. And you can see there's large numbers of records that are housed in many dairy producers' computer records that can be used to actually develop PTAs or prediction equations irrespective of genomics, just with the data they have now, that can be used to start developing high reliability selection for these particular traits. And you can see that when you include the genomics information in there, it gives you even more power, and it improves the accuracies of the reliabilities of these PTAs by about 30%. And so I really see the future as trying to tackle these hard traits, and the development of databases that have this information is going to be really important in that regard. And so one of the things that we've been developing as a result of the BRD cap, and which was in your bags or your goodie bag that you got handed out, is a simplified scoring sheet in order to start recording BRD incidents on commercial dairy farms. And so what we did was we tried to simplify the McGurk scoring system so that it didn't require taking a temperature on every calf, because that's the most difficult observation to get, especially if you're working in California hutches. And so we've got it now where it's just a yes or no. So either the eye is normal, the nasal discharge is normal, or the head, or it's not. And if it's not, then it gets a certain numerical rating. And then if the rating is 5 or more, then it's characterised as an animal that has BRD. And it's both in English on one side and, to the earlier talk, Spanish on the other side. And so I think if we can start getting that records kept, then we can start developing and retraining the prediction equations with new information as time goes on. And so it's really, I think, an important tool. And we're using this now to kind of do benchmarking surveys on dairy farms to have a look at actual rates of respiratory disease. So I'm going to swap gears really quickly and just talk about a survey that we've been conducting as part of the BRD cap, but being conducted by a group in California, particularly led by Betsy Carley, who's a farm advisor in the Northern California, in collaboration with a number of veterinarians. And basically what we did is send out a written survey to all grade A dairies in California and got a snapshot of their management and producer reported prevalence of BRD. And what we're interested in doing ultimately is looking at what the risk factors are associated with BRD. So we started this in the World Ag Expo in 2013, that's about 2 years ago, excuse me, and then did a mail-out survey. Then we sent out a reminder card, then we sent out another reminder card, and we had an incentive. If you filled it out, you were eligible for an iPad to try to get better response rates. And we ultimately ended up with about 15% response rates, which is not too bad for a mail-out survey. And you can see where the dairies are located. So we kind of broke it down into 4 regions of California. The blue is the northern region, which included a lot of the coastal dairies and the northern valley. Then we had the North Valley, which is red shown there. Green is kind of South Valley, and then purple is down in the Chino area. And you can see the average herd size tends to be smaller in the north and get larger as you go down into the valley. And we had a look at what some of their management practices were as it relates to respiratory disease. And so one interesting little demographic was that a very high proportion in the north are raising their own dairy calves, 100% in our particular response. But I was also very surprised at the high proportion of organic dairies that was in the north, and I guess I just want to put that in perspective because as anyone that knows my program knows, I work a lot in the very controversial area of genetically modified organisms, and we recently came out with a paper looking at the prevalence of utilising genetically engineered feed in commercial livestock populations in the United States. And just to put the size of the organic animals segment in perspective, this shows in red the proportion of the different sectors that are associated with organic. And in the case of animal production, it's well under 5% of all animal industries, and in fact milk is the highest proportion that's organic. There's very little in beef broilers and a little bit more in layer hens. But so although there's very high levels there, it's not a huge sector of the dairy industry. So we looked at things like colostrum management, and what we found was the northern dairies tended to leave their calves with the dams longer and often let the calves nurse that dam, which is kind of against what the veterinarian's advice is, which is to try to remove that calf in the first hour of of birth. Typically, they were housed in group maternity pens, and this to the talk about welfare, the majority of the southern dairies were trying to remove those animals within the first hour, and it was much more prevalent in the north that they were staying with those dams 1 hour longer. And so there's this kind of push and pull between the health of the animal and the welfare of the animal, and I think, as with all production systems, there's trade-offs associated with it. Typically, not a large amount of people pasteurising colostrum, and not a lot of people testing it for quality using a colostrometer. So the majority of people were not testing it for quality. And with regards to pre-weaning management, depending on the region, again, there was differences as to whether they were using waste milk, saleable milk, or milk replacer. And of course, waste milk doesn't exist in some cases, like an organic dairy, for example. So you see saleable milk getting used a lot more in the north. Then comes a slide related to the last talk, and that is how much milk or milk replacer is being offered at each feeding. And you can see that almost half of them are somewhere 4 to 5 pints a day, some less than that, some more, but not a great high number there. And of course, some people using medicated milk replacers, others not, depending upon their particular operation. And then the big question we wanted to know is how many animals are getting pneumonia, and the majority reported less than 5%, although there were some that considered that they had much higher rates than that, as high as 25 to 50%. This is producer-reported data, and what we're interested in following up in is having a look at what, if we just go onto a dairy one day and do a spot check, how many of those calves are sick. What's the average age at weaning? In general, somewhere around about 60 days. Some doing very early weaning, less than 35 days. Quite a large number doing much later reading, that is greater than 65 days. And then the thing we were interested in is, well, how are you diagnosing bovine respiratory disease? And the vast majority really didn't have a system or a score. It's like, well, we might look at the eyes, or if it looks depressed, but there wasn't any kind of real systematic way of approaching that. And I hope that this scoring chart might help to get a little bit more precise measurement around that and enable a little bit more of an objective approach to disease recording that. So the next steps is we're doing this 100 farm in-person facility evaluation, and if anyone in the audience is in California and would be interested in participating on that, I'd be happy to put your name on the list. We just do a facility evaluation and then analyse animals to see whether or not they've got respiratory disease. And we're also doing a longitudinal study of 10,000 calves, looking at the overall incidence and prevalence, treatment, and morbidity and mortality data to get a really good handle on how prevalent this disease really is. And as part of that, we're collecting DNA on all of those calves so that we can use that to circle back and have a look how well our genetic tests are predicting the animals that we found that were sick in that particular 10,000 calf study. Ultimately, we'd like to be able to say these are the risk factors that are contributing to increased bovine respiratory disease. And to be honest with you, we've started to look at that data, and the two things that are popping up as significant at the current time are The breed of the animal being Jersey, they have higher rates of bovine respiratory disease as compared to Holsteins, and then group housing is associated with higher rates of BRD. So again, there's that push and pull between welfare of the animal and societal perception of how the animal should be raised, and I think it's an issue we're going to deal with more and more, the trade-offs with these different production systems, and it's just so important to put it in perspective when we consider making changes, what the push and pull is associated with that particular change. So I hope that I've given you an idea that this is a very big project. We're developing these large data sets. I've discussed dairy today. We've also done similar things with beef cattle, and as you can imagine, convincing feedlot owners to go pull healthy controls for a feedlot study is quite a big ask. And our heritability estimates are looking very promising, and so we're hoping that we're going to actually develop some genetic markers or predictions that are going to be useful for predicting susceptibility to BRD. And ultimately, we would like to incorporate this into genetic evaluations so that the PTAs, the normal genetic evaluations, incorporate this trait. And in the long run, the selection of animals that are less susceptible to BRD really offers, a promising and permanent. And once it's happened, you don't have to keep treating it because you've selected for healthier animals, and that's, I think, a goal that aligns with many definitions of sustainability, and of course aligns well with animal welfare, decreased use of antibiotics, and a lot of things that society is pushing for. So I think it's a good story to tell, and we're hoping that we're going to be able to successful to do that. So I just want to finish with, emphasising this is a very large team of researchers located throughout the United States, and it's the effort of all of us working collectively together to get this data that's going to enable these tests to come out. And so I just want to ensure I acknowledge all of the different collaborators that are on that project. And with that, I am happy to take questions and address any things you might have to say.
I wondered if you would share with us what chip you are using. I think you said a high-density 800. Would that be a clarified chip or a gene-seq? Is it available so that other people could try to put information in on, or are you collecting and presenting all those samples?
So we use the 800K chip. It's called the high-density chip. It is not the clarified chip, and it's not the 50K chip. It is a research chip that anyone that wants to could use. It's quite expensive to run it. And really the purpose of it is to hone in on genes and be able to map where exactly that variation is occurring. And so the 50K chip is adequate for what you need for genomic selection. This is more of a research chip in order for us to do gene mapping and the like, and that was the reason we chose it. It's about twice as expensive to run as compared to a 50K chip. We do run it at GeneSeq. They're partners on this project, and it's Illumina is who makes the chip, so it's the same company that makes the 50K chip. But again, it's a research chip that will hopefully allow us to isolate, ideally the gene that's associated with this variation.
The reporting of the disease or the suspicion of the disease, is that something that you're soliciting all of your result, or is this something that some people are actually filling in on their DHIA data and you can get from processing centers, etc.?
At this time, people are not reporting it. So obviously we did our study, but we've put this together, and we've now, with the collection of those 10,000 samples that I mentioned, those dairies are starting to do this scoring system, and we've written a thing to allow it to go into Dairy Comp 305 so that we can get that record associated with the animals. And so if we find that this is a useful tool and that producers like to use this and start recording it, then of course we can start to get a bigger database put together of records. And so this is really the first foray into trying to develop an approach to enable producers to record this health data in an objective fashion so that we can start to develop the data set required for this type of information in the future. Because once we've done our analysis of the 1,000 cows, you know, as generations go by, you need to keep retraining the prediction equations. So we need continuous records. It's not a one-time deal, and that's, I think, what I want to try to set up is a way to enable us to develop those records over time so that we can continuously retrain and make sure that that prediction equation remains accurate in the current generation of dairy cattle.
If it's not information that you can't divulge, would you divulge those 2 specific bulls that you used so that the general population could see, yeah, that's maybe right?
No.
I'd ask.
Yeah, you're not the first person that's asked me that question today. It was really more for illustrative purposes. I mean, it's true data, but it's more for illustrative purposes, and I'd much rather get a prediction equation that we can use for all the bulls, because there's, you know, I don't want to pick on one particular bull who was unfortunate enough to have a bad result in this particular data set. Well. All right, thank you very much.